All right, everyone, we are going to get started on our first chapter in Unit 2. So we're going to cover tissues. Some of this should be a little bit of a review from what we hit in Unit 1, but we're going to go a lot more in depth into our four main tissue types. This one's got a good amount of info, so we'll probably break this into part 1 and part 2. In part 1, we'll cover epithelial and connective tissue, and then part 2, we'll hit muscle and nervous tissue to round out our four main types. Quick shout out to Miss Carlino here for making this great slide deck for you guys. And let's go ahead and get into it. So, what is a tissue? If you remember back from Unit 1, we looked at our 6 levels of organization, and tissue was number 3. It's a group of cells, and this second part is also very important, especially when we talk about our connective tissue. It's not just the group of cells, but it's also the extracellular substances that surround them. And these cells, to be considered a tissue, need to be working towards a common function. So this word hist, or the root hist, is always going to mean tissue. And anytime you see logy, we know it's study. So physiology, we're studying human function. Biology, we're studying life. So anytime you see that word or suffix logy, you know it's going to be the study of something. And in histology, we're studying tissues. And we said this before, we have four basic types of tissue. You have epithelial, connective, nervous, and muscle are our four main types. Glandular tissue is the one that people want to group in there, but remember, as we'll see here shortly, that's a special type of epithelium. And so our tissues are going to be characterized by their structure and function, and truly their structure is what allows them to perform their function. So structure always follows function and vice versa. Before we get any farther into this one, I want to give you guys the reminder, make sure you are using your content guides. So on your course, when you click on your Unit 2 module and scroll to the bottom of your homepage, you're going to see a link for the Unit 2 content guide. That is the best study guide you can have because it's going to tell you exactly how much detail you need to know about the different topics on your test. So I highly recommend having that with you and kind of cross-referencing that as you're going through your recorded lectures and smart books and textbook readings. All right, I kind of just said this, but here's our four tissue types. So epithelial, connective, muscular, and nervous. And so epithelial is our first type that we'll talk about. And this is really covering our body's surfaces. And that doesn't just mean our skin. It's not necessarily all on the outer surface. That includes our hollow organs, our body cavities, and our ducts. And I also said this, but another reminder, our epithelial tissue is going to form our glands. So glands are a special type of epithelial tissue, not their own category. Connective tissue is the other one that we'll talk about a bit more here, and this is going to protect and support our body and organs. This binds our organs together. It's going to store energy reserves as fat. So when you think about our macromolecules, we said carbohydrates are our quick main source of energy, but fat we can also use for energy. And really, it's a great storage reserve for energy. And transport is probably one of the less intuitive ones, but we transport a lot of different hormones, vitamins. We talked about steroids being a fat, a lipid subcategory. So fat is one way that we can transport things, and partly we use fat because our cell membrane is made up of that phospholipid bilayer, which is another kind of subcategory that we looked at with lipids. So it's easier to get fat through fat, if that makes sense. Muscular tissue, we'll talk about our three different types. So all muscular tissue is there for contraction, but depending on where we find it, it has some unique features. The less intuitive one is generating heat. So if you think about when you're cold, what's one of the first things that you do? You shiver because your muscles are trying to warm you back up. Nervous tissue is going to detect changes. So it's a big part of homeostasis. And so it's not only detecting those changes, but then once the that change info gets to the brain, it's going to send out information to our nerves to tell everything else what to do. We have to be able to react to those changes, whether it's with our muscles, glands, hormones, many different things. All right, let's get started with epithelial tissue. And so if you've already had your lab on tissues, a lot of this will probably be a review, which is great because a lot of this info is in your lab manual as well. All right, mostly cells, so very little extracellular matrix. If you see here, there's like no space in between our cells here. So there's no room for extracellular matrix. We just have cells right next to each other. They cover and line our body surfaces, and we also said they form glands. And this term avascular is also important here. So something that's vascular has a really good blood supply. Something that's avascular is going to have no blood supply, really, or very poor blood supply. And that means it's going to rely on everything underneath it, in this case, our connective tissue layer, to feed it its nutrients. So we're going to have diffusion that goes across what we call our basement membrane. So oxygen and other nutrients are going to have to get from the connective tissue across this basement membrane to get to our epithelium. So the main functions, big one is protecting structures from abrasion. So our epithelial tissue is oftentimes the first line of defense in a way. And if you even think about our skin, which is made up of epithelial tissue, we bump on tables and counters and scratch our skin all the time. If we didn't have a bunch of epithelial tissue there, we'd be in trouble. So it's going to protect us from all those little bumps. It also helps prevent water loss, and we'll talk about this a little bit more. But the cells, especially towards the top portion of our skin, especially, is going to form a really good barrier. And so that's going to, one, stop water from getting out, and two, stop microbes like bacteria and viruses from getting in. So it forms a good seal. It does permit diffusion. So we'll see in some areas we have really, really thin membranes, just kind of one small layer of this tissue, and that will allow things to diffuse very easily. And then, of course, we said glands are a special type of epithelium that will secrete substances like sweat and mucus, which have another important role in our body. All right, so whenever we're naming or classifying epithelial tissue, we have to say two parts of the name. So the first part is going to tell us how many layers. So simple, we have one layer. Pseudo-stratified is kind of in between. This one didn't make it to the surface, but this is just one layer. So it doesn't really fit nice in either category. And this is one that you should know for a lecture, but we will not test you on it in your microscope slides. And then we have stratified, meaning we have many layers. So oftentimes, if just like our skin, if we need extra protection, it helps to have a whole lot of layers to do that. The other way they're named is by the cell shape. So you have squamous, so think of squashed, they're flat. Cuboidal are cubes. And then columnar are taller than they are wide, so they kind of look like columns. And so you'll see all of them, no matter what shape or how many layers, have a couple of similar features. So one of those is that basement membrane. And so we looked at that in a previous slide because we said the oxygen and nutrients have to diffuse across this membrane since our epithelial tissue is avascular. The other thing is the top half. We call this our free space or our free edge. So that's the top where it's going to actually face the surface. So for our skin, that's our outer environment. For say the lining of the stomach, that's going to be the inner portion of the stomach. So it's not always facing necessarily to the external environment, but this is facing wherever our space is the free edge or apical edge, and then we have a basement membrane. So no matter what shape, how many layers, those two features are always there. One other one that is a little bit unique, doesn't really fit into these categories, is transitional tissue. And the reason it doesn't really fit into our names is that even though it has a bunch of layers, it kind of changes shape based on what's going on in that tissue. A great example of this would be the bladder. And so when our bladders are empty, this is kind of what our cells would look like. They're more cube shaped. When our bladder is full and the tissue is stretched, these cubes now flatten out because there's more space being taken up there, allowing for that stretch. So anywhere we're having a large change in volume, like I said, the bladder is one of the best ones to remember. That's where we would find transitional epithelium. All right, let's go through it one by one. So here's that free surface that I was telling you about. So that's going to be this top edge. And so in this case, our free surface would be this part that's facing the white opening. You can see the nuclei here in each cell. And then in this case, we have a very thin layer because it's simple squamous. That means one layer of flat cells. So our basement membrane is just this back edge here. So we just have one layer. You can see nucleus, nucleus, nucleus. That's one thin row of flat cells. And so two things, filtration and diffusion is really when we'll find simple squamous because it's only one thin layer. It's very easy for things to pass back and forth. So this is an example from one of the kidney tubules. And so in our kidney, within the tubules, we have a lot of exchange of material. So we will have a thin layer, so it's easy to pass things back and forth. Here's some other locations. This is the one that I was just talking about, but some other locations where we would find simple squamous epithelium. Makes sense. Blood vessels, especially as we get to our really small capillaries where we're going to exchange oxygen and carbon dioxide and other nutrients, we'd want to diffuse across easily. Same thing with the air sacs or the alveoli of the lungs. Carbon dioxide and oxygen are trying to swap really quickly between the alveoli and blood. So these locations should make sense in terms of our functions. So they match up. Okay, simple cuboidal, they're really there for secretion, absorption, and they help with mucus. So I shouldn't say they help with mucus, but they produce and move mucus. Oftentimes, our cuboidal epithelium will have cilia, especially in our respiratory passages or other areas where they're trying to move things along. We also would find these in our kidney tubules. So this is another one. So we see this is a really good quality slide, and we have one layer of cube-shaped cells, and this is zoomed in. So if you're looking at this one, kind of test yourself real quick and see if looking right here, where would be your free edge and where would be your basement membrane? All right, get my cursor back. So this right here, where I'm tracing with my cursor, would be your free edge or your free surface. And then this part here would be the basement membrane. So don't just think top for the free edge and bottom for the basement membrane. Our free edge is going to be facing the open space. So this is a free edge because now we have our hollow opening. This is our basement membrane because it's connecting us to our underlying connective tissue. All right, simple columnar. So now we're still one layer, but we have our taller cells, and we almost always see these drawn with cilia. So cilia or microvilli. Cilia are those hair-like extensions, and they're used for movement of particles. So in our respiratory system, this is really a great example of where we would find these. So our bronchioles. We don't want dust and extra junk getting into our lungs. So the cilia of our simple columnar epithelium can help move that dust or debris or whatever may be in there, either up to get it out of our respiratory passage or down to get rid of it a different way. These, just like our simple cuboidal, are really more responsible for secretion and absorption, and that oftentimes has to do with these microvilli. So those are like little hills on the top of our simple columnar cells, and they're not like cilia where they're moving material as much. What they do is increase surface area. So if you have more surface area, you can secrete or absorb more depending on the location. It gives it more contact with whatever we're secreting or absorbing. And then again, you guys should be studying some of these location examples. And if you really kind of think through your functions, you guys should be able to come up with a couple of guesses on locations just based on what our cells are responsible for. Excuse me. Okay, pseudostratified. So this is the one that I told us to ignore on the lab side of things, but we should be aware of what it is for our lecture stuff. So we don't call it stratified because we have some that only still have one layer, but you can see we have a couple that get kind of stuck and don't actually make it all the way to that free surface. And so we're always going to have our cells attached to the basement membrane. So that's the way it connects to the underlying tissue and gets its nutrients. And these are really there to make and secrete mucus. So nasal cavity, nasal sinuses, our trachea, which is your windpipe. That's our main kind of start of our air before we split into the bronchi to the lungs. So we find pseudostratified columnar and some of the same places we find simple columnar. So this one, definitely think respiratory. All of these are respiratory system examples. Same thing with the cilia. It's trying to move mucus with those extra extensions. All right, stratified squamous. So now we're getting into our multiple layers. So that could be 2 layers, that could be 2,000 layers. And so you'll see that really the flattest cells are at the top. Once we get down lower, they actually are a little bit more cuboidal or columnar looking, but you're really looking up here in terms of knowing these are stratified squamous cells. And part of this is to protect from abrasion and that water loss. So this top layer, we have a lot of extra cells that if these guys get bumped off, we have a lot that can take their place. And so I'll mention this now. One thing you can see here is that our top layer really doesn't have any nuclei left. We see one or two here, but we see a lot of just white. And that's because as our stratified squamous epithelium gets pushed up from the basement membrane, it gets flatter and it fills with keratin. And so that keratin is a hard substance that is what allows this top layer of our stratified squamous epithelium to form such a good seal. And so our ones down here on our basement membrane, this is where our cells are actively dividing. So this is where we're getting our new stratified squamous epithelium from. And as we divide from this bottom layer, one of our two daughter cells is going to push up. And so eventually we get to where they flatten out and we get our protective layer. We'll talk about that a little bit more. So areas of friction or that are going to have an in abrasion often are really where we're going to find these. So again, these should make sense based on our function. All right, we really don't talk much about stratified cuboidal or stratified columnar epithelium because we really don't find it much. So make sure, oops, sorry guys, make sure you take a look at our functions and our locations here for a couple of unique examples, like sweat glands and salivary glands are a good one to know for this stratified cuboidal. But these are rare enough that we really mostly only talk about stratified squamous. Okay, transitional. So we talked about this briefly earlier. It's not quite fitting into our simple or stratified category. Um, it's clearly stratified, but because it doesn't maintain the same shape, we call it transitional. And so our cuboidal cells are when there's not a stretch. So they look like cubes when we're empty. When they're full, we need more space, so we kind of have to squish our cells and they become more of a squamous shape. So like I said, the best example, think urinary bladder. The ureters and portions of the urethra also do this, and same thing, it's accommodating that change in volume. So we're going to have a difference when there's no fluid versus full. All right. Now we're going to talk about cellular connections or how our epithelial cells stay connected because we said there's very little space between our epithelial cells, and this is why. So some of these mechanically bind cells, you'll see, we'll talk about some extensions that almost act like screws or nails would for lumber or a wall to hold pieces together. They also help form a barrier sometimes. And this is another way that our cells communicate. So this is more almost like extensions that tell us what's going on in this cell, and it can tell this cell what's going on, and vice versa. So we'll talk about all these different examples here. All right, our first one is desmosomes. And so this is one of our mechanically binding. So you can see we have these intermediate filaments and what's actually kind of a glue or an adhesive material between the cells. So it kind of glues the two cells together, and then it has these intermediate filaments that are going to extend into the cytoplasm of both cells. So it helps keep things really tight together, but we also have structures that can communicate a little bit. But really, this is just here to hold things tight. All right, hemidesmosomes. It's a half hemi of a desmosome. And so these are going to be for our basement membrane. Oops, sorry. I was trying to highlight, but I clicked to the next one. So this one is going to go kind of in the middle. If we go back to our big picture, two cells together. Our hemidesmosome isn't connecting our two cells, it's connecting our middle cell to the basement membrane. So that's the big thing to remember. Hemidesmosome only has half, so it's just going to the basement. All right, tight junctions do exactly what they say. They form a tight barrier, and the whole idea is that it's going to prevent molecules moving between cells. We've talked about this a little bit with diffusion and concentration gradients and even the sodium-potassium pump. We want certain molecules to stay inside a cell or outside of a cell. Some stuff we need to go back and forth, but a lot of times there's stuff that we just want to keep out or keep in its individual cell. So these tight junctions are going to prevent movement of molecules that shouldn't be moving. They also help anchor cells to each other. So just like our desmosomes, you can see we have extensions on either cell, so it's connecting them. And these are typically found more towards the free surface. So we had the hemidesmosomes down by the basement membrane. Now we have our tight junction that's higher up towards the free surface. We find these primarily in the stomach and urinary bladder are the two examples that I want you to remember for this one. All right, gap junctions are a little bit different. These are actually protein channels, and so you can see protein channels, and this is what's going to allow small molecules and ions to move between cells. So the channel is going to be specific to the molecule or ion, for example, a sodium channel or a potassium channel. And so this is what actually allows our nerve and muscle impulses to travel between cells really quickly. If they had to go all the way across the membrane without those channels, or if we had to tell each individual cell the same message, it would take a lot longer. It would be really inefficient. So this is how those ions or that signal can pass a lot quicker. So most of the ones we've talked about are trying to kind of keep things out or hold things together. This is more for communication and to allow signals to travel quickly. So one example of this is our nerve cells. We'll talk about these a lot in our nerve cells, actually. But the other one that we mentioned, if you've had lab, is intercalated discs. And so we'll talk about these again when we hit cardiac muscle later on in this lecture. But our intercalated discs are a combination of gap junctions and desmosomes to keep things as tight as possible, but also let that nerve signal that's causing our heart to beat to pass quickly in between our different cardiac muscle cells. All right, last thing here for epithelial, we have the glands. And so these are specialized secretory organs of epithelium, and they're going to support a network of connective tissue. And so I'm sorry, they have a supporting network. They are not the ones supporting. Let me make that clear. So we said our epithelial tissue is avascular, so it doesn't necessarily have anything to supply our glands with. So it needs to get its material from that connective tissue underneath. We have two main types. Endocrine are going to go directly into the bloodstream most of the time. They don't get to the exterior. They don't have an extra duct. They just secrete their hormone right into the bloodstream. Whoa. Our exocrine glands are going to secrete products into ducts, and they're going to empty onto the surface of epithelium. And again, remember when we say the surface of our epithelium, we're talking about that free edge. It's not always going to be the external environment. This could be internal, just like the mucus secretions in our digestive tract. But the big difference here is that they have ducts. The exocrine glands have to exit through a duct, and they're going to go onto the surface of our epithelium. So we have a few different examples of these: our sweat glands, our oil glands or sebaceous glands, our earwax, which is cerumen, and then, of course, saliva and mucus. Okay, just to keep it easy, I'm going to go ahead and end this recording for now, and we'll pick back up with connective tissue, which is probably our largest and most broad category to talk about.